On today's Leading Edge, we look at the life and death of cells. Taken at cell level, our bodies consist of cells that are dying and being born all the time. What does the latest research tell us about this process?
Using bioimaging techniques, which we have covered before on Science View, it is now possible to observe cellular processes. This revolutionary technology allows scientists to light up living cells for observation. Using this technique, the once hidden drama of the life and death of cells is being revealed to science.
First, let's look at some research that has provided a new view of cell death.
Apoptosis is a process whereby cells kill themselves. For new cells to be created, it is necessary for damaged cells to destroy themselves in a programmed cell death. Professor Masayuki Miura of the University of Tokyo Graduate School of Pharmaceutical Sciences is studying cell apoptosis. Along the way, his research ran into a major obstacle.
Cells that consume dead cells are constantly on the lookout. They immediately engulf and digest any dead cell, making it very hard to spot the moment of death.
Here we see an immune cell called a macrophage engulfing a cell that has just died. Dead cells are gobbled up and digested by macrophages like this right away, making it very difficult to observe cell death. So Miura came up with a strategy. When a stress is applied that triggers apoptosis, a chain reaction leading to cell death is initiated. Along the way, there is one substance that always springs into action, the enzyme caspase. Caspase has two main functions. It acts directly to sever the cytoskeleton, and it stimulates a different enzyme to snip apart DNA strands. These two functions are essential to apoptosis. Miura developed a fluorescent protein. The protein changes its color. As caspase is activated, the protein turns from red to blue. He then used it to observe the epidermis of a fruit fly. First, he imaged this part of the pupa. Appearing in red are cells of the pupa. Gradually, they change to green and then to blue. The changes in color demonstrate how caspase is activated, inducing cell death within the pupa. Next, Miura imaged a larger area. The darker areas are the epidermis of the adult fly, and the lighter colored that of the larva. The caspase activity is most pronounced at the boundary between them. On the screen, moving from left to right, the larval cells are successively undergoing apoptosis and being replaced by adult epidermis. Miura successfully imaged apoptosis in real time. This remarkable metamorphosis from larva to adult is dependent on programmed cell death.
This was the first time we could observe caspase being activated according to a specific pattern or from a specific place, and the dynamic process of cell death that ensues. Seeing vivid images of the body carrying out this systematic program of cell death and replacement with new tissue was a powerful experience.
By capturing footage of apoptosis in vivo, Miura also discovered that the body uses a process of trial and error to build itself. Along a fruit fly's back are bristles spaced at regular intervals. These tell the fruit fly when something has touched it. How is the even spacing of these external sensory organs achieved? Until recently, it was thought that precursor cells, which form the external sensory organs, are evenly spaced. Because no new precursor cells can be formed around these cells, the external sensory organs naturally come out evenly spaced. But Miura's observations overturned that idea. The white dots are newly formed precursor cells. They are clearly not evenly spaced. In places, several cells are clustered closely together. Now apoptosis occurs. Let's take another look. These 4 cells vanish. Many precursor cells compete. The external sensory organs form above the cells that win out. The orderly arrangement of external sensory organs is the end product of this trial and error process.
When the neuronal precursor cells form, they're competing with each other. It's survival of the fittest. As all of these cells fight for survival inside of the pupa, we see the drama of life unfolding. This is an extremely dynamic stage, but it is invisible from the outside.
Build and destroy, destroy to build. This dynamic process generates a great diversity of living things.
That's right. It's a survival of the fittest among the cells of the body, with the fittest remaining and the others destroying themselves through apoptosis.
But isn't it wasteful to destroy cells that the body already made?
Even in brain development, first, a large number of synapses are created to act as links between neurons. Then, a culling process takes place in which only the synapses required to enable the neurons to communicate information are left. So, making a surplus first and selecting only the necessary ones after is a rational process.
We've been looking at apoptosis, which is necessary for forming the body. There's a more sudden and destructive type of cell death as well.
Yes, there is. Please look at this. These are mitochondria. They are crucial to the cell, supplying it with energy. But in doing so, they generate molecules called free radicals. The most powerful type of free radical is called hydroxyl. Hydroxyl radicals savagely attack cells, causing cell death.
How does cell death triggered by mitochondria happen? Let's watch.
This is Shigeo Ota, professor at the Nippon Medical School. Ota developed a technique for making parts of a cell where hydroxyl radicals are generated appear red. In the center is the nucleus, surrounded by mitochondria shown in yellow. These mitochondria have just released hydroxyl radicals. Soon afterward, the mitochondria in this area turn blue. This shows that the mitochondria have been oxidized by the hydroxyl radicals, resulting in strong acidity that destroys them. Now, hydroxyl radicals are released even more widely. After that, within 3 minutes or so, all the mitochondria have been destroyed and the cell dies.
This is the first time we have seen how fast mitochondria can destroy a cell. It's very fast. Unlike apoptosis, this is what you might call an accidental death, one that occurs in a brief instant. If this sudden death occurs over a wide area, it could damage the entire organ or lead to the death of the whole organism.
Mitochondria constantly work to generate energy for the cell, but sometimes release powerful free radicals that destroy the cell. These contradictory aspects of mitochondria come from a remarkable evolutionary history. Researchers believe that until around 2 billion years ago, mitochondria were independent organisms. Then at some point, they were engulfed by larger cells. The DNA of the host cell and the mitochondria were stored together in the cell nucleus. The mitochondria produced energy and the cell protected the DNA. In this way, there was a symbiotic division of labor. This arrangement has continued to the present. Here we can see where ATP produced by mitochondria is distributed in the cell. The area in red indicating high concentration of ATP is the nucleus. But the mitochondria appear green, showing a lower concentration of ATP. Cells co-opted mitochondria as a source of energy, but in doing so, introduced a potentially fatal force within themselves.
So it's completely up to the mitochondria whether the cell lives or dies?
Not really. The cell has mechanisms to decide on cell death if the mitochondria begin to malfunction, for example. Mitochondria and cells are interdependent, yet there's also a certain tension in their relationship.
Thanks to bioimaging, we now know that life and death for cells are like two sides of the same coin. Let's move to our next topic on the life of cells. Germ cells are the cells that will produce the next generation of an organism. But at what stage do they form? Let's take a look at research that is solving that mystery.
One researcher's observations of killifish has captured the moment when germ cells are born. This is Associate Professor Minoru Tanaka of the National Institute for Basic Biology. He's not interested in the adult killifish, but the eggs slung beneath it. Why did Tanaka choose to focus his research on this species of fish?
The eggs of the killifish are transparent, so we can watch the embryo grow and develop until the eggs hatch.
Here is a time-lapse view of a fertilized egg. On the left of the screen, cell division begins. From fertilization to hatching takes this species 10 days. This is still only the 10th hour. The body forms from a cloud-like clump of cells called the germinal disc. At this stage, the germ cells are already emerging. Tanaka treated an egg with fluorescent proteins and filmed it from the direction shown by the arrow. Let's watch. Did you see the bright dots? This is the first ever footage of the moment of germ cell genesis. Here are the germ cells at greater magnification. They have emerged just 10 hours after fertilization. Germ cells in the killifish form even before the body.
To create the next generation, a cell must be able to differentiate into all the cells of the body. We say it must be pluripotent. In order to retain pluripotency, the cells must be set aside at an early stage before differentiation. That is probably why germ cells are handled in a special way at this early stage.
But creating the germ cells so early creates a different problem. Let's look again at the germinal disc at that time. There is not even a trace of the testes or ovaries that would house the germ cells. How do the germ cells handle this situation? The next video was taken from this direction. Newly created germ cells migrate to the margin of the germinal disc. At higher magnification, the germ cells can be seen creeping along under their own power like amoebas.
What we are seeing here is the germ cells after they emerge migrating toward the gonads, toward the tissues that will become the ovaries or testes.
1 day and 17 hours after conception, the axis of the body, which will become the spine, has formed. The eyes also begin to form. Meanwhile, the germ cells are migrating down the axis of the body. Moving forward about 2 days, to the 4th day after fertilization, the germ cells have arrived. Here, germ cells are shown in red, and cells that will develop into the testes or ovaries are in green. Having completed their long journey and finally arrived at their resting place, the germ cells await hatching. Here they will remain until the organism reaches sexual maturity.
Once the body has formed to the point where it can produce offspring, the germ cells finally become active again. In that sense, from the viewpoint of the germ cells, our entire bodies could be considered a kind of box or vessel which is designed specifically to carry germ cells.
Given that germ cells are only active in adulthood, they are created at a very early point.
That's right. In humans, the germ cells appear around 3 or 4 weeks after conception. After that, it is thought that they undergo various stages of apoptosis in a process of careful cell management, which is aimed at ensuring we can produce offspring. Actually, the way the number of cells is regulated is not yet understood. Hopefully, the bioimaging revolution will unlock this secret of life as well.